Okay, we're very excited to have with us today Daniel Veronese-Paniagua. He is coming to us from the Millman Laboratory at WashU St. Louis School of Medicine. And his talk is investigating the effects of coxsackievirus B3 infection in the various islet cell types. A little bit about him. He's a Venezuelan stem cell biologist with a keen interest in understanding the induction of type 1 diabetes pathology and advancing medicine through the development of cell therapies. He's co-authored multiple peer-reviewed publications in high-impact journals, including Stem Cell Reports, Cell and Molecular Gastroenterology and Hepatology, AJP, Gastroenterology and Liver Physiology, Nature Cell Biology, and Nature Communications. In 2019, he began his PhD in developmental regenerative stem cell biology at WashU St. Louis. As a graduate researcher in Dr. Jeffrey Millman's lab, he's investigated the role of genetic variants in the onset of type 1 diabetes under various environmental triggers. In leveraging his expertise in single cell RNA sequencing, he has also explored the effects of coxsackie B3 infection on distinct cell populations within human pancreatic islets. His academic achievements and contributions to research have been recognized with numerous honors, including the Nancy Horton Bartels 48 Scholar Fellowship, an NSF Research Experiences for Undergraduate Internship Initiative for Maximizing Student Development Training Award, and an award from the NSF Graduate Research Fellowship Program, and an outstanding poster research presentation at SACNAS, DI STEM Annual Conference, and an internship with Lilly. So Daniel or Danny, we are very happy to have you here today, and we're really looking forward to this presentation. I'm going to drop your recent preprint into the chat for people's reference, and yeah, take it away.
Great. Thanks so much, Monica, for that introduction. And thank you all for attending my talk today on the effects of coxsackie-virus B3 infection on the various islet cell types. So I'm just going to start with a little bit of background that I'm sure many of you are familiar with, but diabetes is a chronic disease that is currently affecting about 537 million people worldwide. And it's actually going to be increasing in the next few years in all continents of the world. And it's projected to impact about 643 million people by 2030. So all forms of diabetes are associated with the pancreas, and within the pancreas, we have these heterogeneous tissues called the islets of Langerhans, where we have the somatostatin secreting delta cells, pancreatic polypeptide secreting gamma cells, glucagon secreting alpha cells, and then the insulin secreting beta cells. And together, all these cell types work to regulate blood glucose levels, mainly by the regulation and secretion of insulin. Now, in type 1 diabetes, we have an autoimmune attack that targets and destroys the beta cells, and essentially leading to loss of beta cells. And also, there's literature showing dysfunction of alpha cells. But all of this leads to decreased insulin secretion, and as a result, we have increased levels of sugar in the blood. And there's been research showing that the autoimmune attack is caused in part by autoantigen production by beta cells. And I just want to have this slide here summarizing the different causes that induce autoantigen production in beta cells. And they can all be summarized by just saying that beta cell stress can induce production. And beta cell stress can include ER stress, mitochondrial stress, exposure to cytokines by the cell, and also viral infection. So with this in mind, many groups have sought to identify and investigate the triggers for type 1 diabetes. So for decades, it's been known that type 1 diabetes is a genetic disorder and that there are multiple hundreds of SNPs associated with T1D. And here I'm just highlighting some of them, including the HLA, as well as genes associated with the PI3K pathway and Notch signaling and type 1 interferons. But what the field has also shown is that T1D is a discordant disease. And what this means is that when we look at identical twin studies, only about 65% of twins go on to develop type 1 diabetes, and 35% do not. And you just got to remember that identical twins share the same genetic background. And so this was a very surprising find. And with that in mind, the concept is, well, genetics alone cannot explain the initiation and progression of type 1 diabetes. And there's been a search for what are the other factors that are involved in initiation and progression of T1D. And many groups have turned their heads towards environmental factors. And some of these can include things like low microbial diversity, diet, which can also affect the microbiome. And I know the microbiome has gotten a lot of attention in the last few years. Also, low levels of vitamin D have been associated with T1D, as well as exposures to toxins and chemicals. But there's also rising evidence that viral infections can be a potential trigger to T1D. And this is actually supported by the genetics of T1D because there are multiple genes involved in the antiviral response, such as IFIH1 and TIC2. If we narrow in a little bit more into this topic of viral infections, there's actually been multiple studies that have highlighted a correlation between coxsackie B infections and type 1 diabetes. And so if we go into the pathology of Coxsackie, the way that it works is that the virus binds to a surface receptor called CD55 on the cell. When it binds, this induces CD55 clustering, and as a result, Coxsackie can actually go on to bind the CARB surface receptor, which will then help the virus be endocytosed into the cell. Once it's in the cell, it will be replicated, its genome will be replicated, and virions will be produced to then eventually be released by the cell and expand the infection. However, while all of this is happening, there's also a double-stranded RNA called MDA5 that is encoded by IFIH1. And MDA5 actually is able to detect the double-stranded RNA from Coxsackie and other viruses. And once it does this, it will bind to the MAPS protein on the surface of the mitochondria. And together, they will induce type 1 interferon signaling and subsequent immune responses in the cell. Other studies on Coxsackie virus, mostly on cancer cell lines, have also shown the specific effects of the virus on multiple cellular organelles, including the Golgi, the endoplasmic reticulum, the mitochondria, and autophagosomes. And interestingly, all of these organelles are also involved in cell stress. And if you remember the slide that I discussed before, cell stress in beta cells is usually associated with a production of autoantigens. And so this is definitely something to keep in mind for this talk. Studies on Coxsackie virus and islets have shown that here is just some histology images or microscopy images, sorry, but essentially infecting primary cadaveric islets with Coxsackie virus B4 induces cell death in these islets after 4.5 days compared to controls. The same study also looked at the effects of different types of Coxsackie viruses on function of the primary islet. And basically, here in white is the control, and the other bars are the primary islets infected with viruses. And I just want to show that all the Coxsackie virus type B, such as CVB4, 5, and 3, are able to reduce the insulin secretion in these cells. However, the serotype A of Coxsackie virus is not, and this is represented here by these bars with a straight line. Also, another study, what they did was that they transplanted primary human islets into the kidney capsule of NOD mice, and then they infected the mice for about 40 to 50 days. And what they showed is that on average, the mice are actually able to develop type 1 diabetes about 28 days after infection. So these studies have focused on the effects of islets on, the effects of virus infection on whole islets, but there have also been some studies on the impacts on individual cell types within the islets. And so this group isolated rat alpha and beta cells and then infected them with Coxsackie virus as well. And here the dotted line is just the mRNA expression of these different type 1 interferon genes normalized to the beta cell. And then the bars actually represent the expression of these same genes in alpha cells. And what they showed is that alpha cells have a stronger immune response in response to Coxsackie virus infection. So if we look at other areas of the pancreas, there was another study that infected cell lines with Coxsackie virus, did bulk RNA sequencing, and what they showed was basically change in gene expression associated with lysosomes, extracellular matrix, and immune response. But despite all this, a lot of the studies so far have focused on the holistic view of primary human islets, as well as individual cell types derived either from rodent models or from immortalized cell lines. So the effects of coxsackie-virus infection on specific cell types in whole human islets remain unknown. And that's kind of like where my paper comes in, which is currently under review with Cell Reports. But the goal of the study was to characterize the cell type specific transcriptional responses to coxsackie-virus in human primary islets. And the hypothesis for that study was that infection induces unique cell type specific transcriptional responses, as you can see here in this diagram, where each cell responds differently to the virus. And the way that we did this was essentially we took cadaveric islets from 4 different donors, and then we exposed them to different treatments for 24 and 48 hours. And these treatments include a negative control, which was just endotoxin-free water, a positive control, which was a synthetic double-stranded RNA called poly-IC that induces type 1 interferon signaling in cells, and then finally, coxsackie-virus B3 fused to EGFP. And then after the treatment, we went on ahead and sequenced these cells using 10x single-cell RNA sequencing technologies, and also collected cells for other experiments. Here, it's just a qPCR, basically showing that we were able to successfully infect the cells prior to sequencing. And so here in the gray is control, blue is the poly-IC, and the green is the coxsackie infected whole primary islets, 24 hours and 48 hours. When we go on ahead and start our Seurat analysis, essentially what we were able to show the presence of different cell types within the pancreas. And here on the right side, it's just a heat map for different cell markers that correspond to the clusters here on the left. And so what we can appreciate is that the primary islets not only have the different endocrine cells present in the islets, but also we have some exocrine tissue in the form of ductal cells and acinar cells, as well as other cell types, including endothelial cells and monocytes. So if we start from big picture with our analysis, and we look at first at what's been done in the past before in terms of type 1 interferon signatures, and look at it in the whole islet, we show that at 24 hours, there's some robust increase in different type 1 interferon-associated genes when the whole islets are treated with both poly-AC and coxsackie-virus. What was really cool to see, too, is by 48 hours, poly-AC, as expected, further increases the expression of these genes, but coxsackie-virus actually decreases the expression of these genes. And you may ask yourself why. Well, if you think about it in terms of viral infection, viruses don't want to be detected by the immune response. So this wasn't too surprising in a way, because coxsackie-virus will enter the cell and then it will try to hide itself from the immune response so that it could keep infecting the cells. And if the cells are telling the immune system, hey, there's something wrong with me, then that kind of goes against evolutionary patterns there. The next thing that we did is that we also looked at the differentially expressed genes of both treatments compared to control. And what we saw that was very drastic was the difference in numbers of differentially expressed genes when comparing poly-IC to coxsackie-virus at both 24 and 48 hours of expression.
Daniel,
And
I have a quick question from
yeah.
the chat from Larry Spears. The
Mhm.
effect of CVB3 transitory, does the pancreas recover? Do they create a sustainable signature that can be looked at in the population and maybe treated with antivirals?
Yeah, that's a great question. So our study only focused on 48 hours of infection at most, but there have been some studies trying to look at the long-term effects of virus infections and whether coxsackie-virus and other members of its family are capable of infecting tissues long-term. And at least in the pancreas, there have been some studies that have shown low levels of viral proteins and also viral genomes measured through PCR. So the virus is capable of staying in the pancreas for a long period of time. But whether it has negative effects either cyclically or chronically, over time is still understudied. I think that right now it's really hard to do that with ex vivo primary tissue because we can only culture the islets for about 2 weeks before they start disintegrating and dying. And then with mouse models, we do see that the islets are negatively affected after about 28 days, but there haven't been models showing this for longer periods of time.
So wait, can you just clarify, is this CVB reminiscent of the Epstein-Barr virus where it kind of infects and then can be quiescent for a while and then get reactivated? Is that what you're saying, or there's not enough information to even say that?
There's not enough information to say that. What I can say is that we can detect the viral RNA in both stool samples and also through PCR of cadaveric pancreas after months after the infection. So, but whether it reactivates and over time is still unknown.
Okay, thank you very much.
Mm-hmm. Are there any other questions?
Not yet. I'll
Great.
let you know.
Thank you. So when we look at the shared genes between the two treatments, here in red are genes that are shared between them and also upregulated in both. And a lot of these genes are associated with type 1 interferon. We also did see differences in the way in which the genes were expressed. So here in black are some genes that are upregulated in coxsackievirus, but downregulated in poly-IC. And some of these include components of tight junctions, such as CLDN7, as well as modulators of cell signaling, such as PEB1, which modulates MAP kinase and NF-kappa B. But basically, when we look at the cell type specific effects of poly-IC compared to coxsackieviruses, a lot of the signatures are very similar. They're all just, for the most part, differentially regulating genes associated with type 1 interferons. And then coxsackievirus has a more dynamic response. So because of that, in the rest of the talk, I'm just going to focus on comparisons between coxsackievirus and their respective controls.
Quick question in the chat,
Yeah.
Danny, is from Mehdi Bancala, San Diego. Maybe I missed this, but what was the concentration of CVB used to infect the islets?
Yeah, so the MOI of CVB to infect the islets was an MOI of 20, which I recognize that that's a high MOI, but it was necessary because we were working with whole aggregated islets. And so these islets were cultured in suspension. For example, if we take the same islets and we single cell disperse them, we have to reduce the MOI to about 0.1 for us to be able to perform assays. But for us to get a robust immune response in these cells, we had to increase the MOI to 20.
Great, thank you.
Yeah. So when we look at the infection rates across the different cell types in the whole islet through single-cell RNA sequencing, we can actually see that there's similar infection rates of coxsackievirus across all the different cell types. And this was very interesting because here on the right side, I have the expression of the entry receptors in these different cells. And we can actually see that only delta cells and ductal cells co-express at high levels both of the entry receptors. There is some expression of CXADR, which encodes the CAR surface protein in the other cells, but it's just a lot higher in ductal cells and delta cells. So despite similar infection rates, we actually see differences in transcriptional response across these cells, with alpha cells, beta cells, and then ductal cells having the strongest transcriptional response in terms of the number of differentially expressed genes. So this was really neat to see, but obviously just having a list of genes isn't enough information. So we took it a step further and we performed independent gene ontology analyses for each cell type at both time points. And then to get a little bit more information out of gene ontology, we categorized statistically significant gene ontology terms into groups that describe various processes and structures, such as gene expression, mitochondria, vesicles, and immune response. And we dubbed these groups as GO themes. The thing that we did next is we took the list of genes that were present in these GO terms, and essentially we ran pairwise comparisons for each cell type, comparing the corresponding control to the corresponding infected cells. And then we determined the number of differentially expressed genes with statistically significant fold changes. And that's where we're able to get this bar graph here for the different gene ontology themes. And essentially what we show is that, as expected, immune response associated genes are have the highest number of significant expression, followed by protein metabolism and trafficking, as well as vesicles and mitochondria. So because we're talking about viral infections, I'm going to focus a bit on immune response in this next slide. And so I'm going to walk you through this heat map here, but the big picture from from both heat maps is essentially that we can see that there are different patterns to gene expression within the alpha cells, beta cells, and ductal cells. And so, for example, at 24 hours, we see this block of upregulated genes and beta cells that is not present in the other cell types. And some of these genes include type 1 associated genes such as RAS-GRP1 and PTPRN2. We also see, for example, some upregulation in both alpha cells and ductal cells in some stress genes such as XBP1 and RF9. And then in beta cells and ductal cells, we also see increased expression of other stress genes such as HSPA5 and LY6E. At 48 hours, we do see differences. Beta cells, for the most part, decrease a lot of immune-associated genes, whereas alpha cells have some reductions and some increases as well. But what was very neat about the 48-hour data set is that alpha cells and ductal cells mostly increase genes associated with type 1 interferon. So in alpha cells, we have STAT3, RRF1, and then with ductal cells, we have ISG15, IFIT1, IRF7, and more. And so what this data set basically showed us is that alpha cells and ductal cells actually have a stronger immune response to coxsackievirus compared to beta cells. The other thing too that we noticed at both time points is that many of the genes differentially expressed in alpha cells and beta cells are associated with ubiquitin components and products involved in protein degradation and the unfolded protein response. So because of that, we also evaluated the gene ontology themes associated with vesicles, lysosomes, and autophagy. And here I won't focus too much on the genes, but more so the overall patterns, where we see that at 24 hours, the beta cells have a very strong increase in genes associated with vesicles. And by 48 hours, we see the opposite, where we see a reduction of vesicle-associated genes. And then when we look at alpha cells, we see very little changes in vesicle genes at 24 hours, but then a more drastic response by 48 hours. And vesicles was very interesting because if we go back to the data where I showed similar infections rates, despite differences in expression of co-receptors, there's been a lot of studies done on HeLa cells and other immortalized cell lines that have shown that coxsackieviruses can actually use vesicles and exosomes to infect other cell types that do not co-express their N2 receptors. So this transcriptional data may hint at a potential infection mechanism by coxsackievirus into these other cell types. When we look at genes associated with autophagy, we basically at 24 hours see that all the genes associated with autophagy are increased in only beta cells and monocytes, with the exception of FTL here in the blue. And then by 48 hours, we see major reduction of autophagy-associated genes in beta cells, as well in alpha cells and the mesenchyme, and no statistically significant differential expression in monocytes. So why is autophagy important? There's also been some literature out there in HeLa cells and acinar cells showing that autophagy is actually used by coxsackieviruses to replicate and to release virions into the microenvironment of the cells. So another GO thing that we also look to kind of to change course here into some more functional data based on our transcriptional data is we also looked at the mitochondria. And this hit was really interesting to us because endocrine cells in general are very metabolically active. And when we looked at the data set for the 24 and 48 hours, we showed that alpha cells, beta cells, and ductal cells actually have a lot of differentially expressed genes associated with the electron transport chain. And it's different components of the electron transport chain. We see changes in component 1, 4, 3, and so on and so forth. And this was interesting because there's some literature showing that other viruses, including HIV, HCV, and HBV, actually hijack the electron transport chain within mitochondria to promote replication and survival. And so because of all of this and our data set from single-cell RNA sequencing, we decided to perform some functional studies. So first, we measured the levels of reactive oxygen species in whole islets infected with coxsackievirus and also treated with poly-IC for 48 hours. Here, the NAC is an antioxidant that serves as a negative control, whereas entamycin A is a positive control. And so what we can see is that coxsackievirus does induce increased reactive oxygen species levels in whole islets significantly. The other data, the other experiment that we performed was also we measure respiration by our whole islets using seahorse. And we took this method from Yepes et al. 2018, where essentially what I want to draw your attention to is one, the dotted line here corresponds to levels for control. We also, if we look at the EAI, which corresponds to proton leak, and then EI, which corresponds to basal respiration, and then finally NEI, we actually see that proton leak in coxsackieviruses is increased relative to control, as well as in poly-IC, but basal respiration and maximal respiration are actually decreased following infection compared to control. And that is not something that we see in poly-IC treated cells. So what does this mean? What does it mean to have an increase in proton leak and a decrease in basal respiration and maximal respiration? Well, according to Yepes, whenever we see that in their using their method, it actually hints at mitochondrial dysfunction, which is validated with this ROS data that I showed before. So we went on also, we were interested in the effects of virus infection on the morphology of the mitochondria, since we see that it negatively impacted the function of it. And so we took the primary islets and then imaged them using transmission electron microscopy. And these are some neat images from beta cells at 24 hours for control and infection. When we quantified the area of the mitochondria, I just have a little bit of lag here. Oh, sorry.
That's okay. Are you gonna? It's not progressing.
Yeah, it's not progressing on my screen. Oh, there we go. Okay, so when we quantify the mitochondrial area in alpha cells, we actually see that with infection over time, the size of the mitochondria decreases compared to controls. And in beta cells, we see the opposite, where infection induces the increase of mitochondrial area in these cells. And this was really nice to see because we not only showed that transcriptionally we have cell type specific responses, but also morphologically in the mitochondria, we also see the same thing. So when we moved on to the beta cells and alpha cells and doing a deeper dive into the analysis, we reclustered the cells to investigate any dynamic molecular states during viral infection. The other thing too that I did was we essentially divided the cells into 3 different groups. One of them is called the bystander cells, which are basically cells that are present in the infected microenvironment but do not have virus itself inside of them. Then we have low GFP cells, which are basically cells that contain less than a 50 percentile of CVB EGFP mRNA, and then finally high GFP cells, which are cells that were in the upper 50th percentile of the viral titer. And when we perform Pearson correlation, we actually see that in beta cells, the bystander cells are very transcriptionally similar to the high GFP cells, which are the ones that have high viral titer, which is kind of surprising to us because we actually expected something more like what we see here on the right for alpha cells, where bystander cells are more transcriptionally correlated with control cells. When we focus just on the beta cells and look at the genes, the top upregulated genes, we can see the differences between the control, the bystander, the low GFP, and the high GFP, where there's some overlap between infected cells and with these genes, and not so much for bystander. So for control, we see increased expression of a lot of beta cell identity genes. For bystander cells, it's mostly genes associated with identity, but also function. And then for low GFP and high GFP is a lot of cells, a lot of genes associated with stress response. And one of these genes that caught our attention is actually this long non-coding RNA called miR7-3HG, because it's been very understudied in the context of the pancreas. And when we check the expression of miR7 in our patients through qPCR, we do validate our single cell data where we see increased expressions of miR7 after viral infection. So to study miR7, we leveraged our human pluripotent stem cell differentiation protocol, where we take human pluripotent stem cells and expose them to different drugs and factors to mimic pancreatic development. And that helps us generate stem cell-derived pancreatic islets. So we took these SC islets and then infected them with either lentiviruses containing short hairpin RNAs against GFP as a control, and then against miR7 to knock down miR7. And here on the right side, it's just some qPCR showing that we're capable of knocking down miR7-3HG in these islets following infection with two different shRNA sequences. And when we look at the viral genome expression in these islets after coxsackievirus infection, we obviously, compared to control, we see the increase in viral genome. But then in the miR7-3HG knockdown, we actually see reductions of the viral genome compared to the wild type, which was really neat. And the other thing too that we noticed is decreased expressions between the knockdown and the wild type following infection in DDIT3, which is a stress marker that is usually associated with apoptosis. And so with all of this data, when we decided to do some functional assays on the apoptosis, we actually see reductions in the apoptosis rate in our stem cell-derived islets with miR7 knockdown compared to wild type with both of these infected with coxsackievirus. So in conclusion, I just want to say that we showed that the multiple islet cell populations have unique cell type and condition-specific transcriptional responses. I also showed that alpha, beta, and ductal cells have the strongest responses to coxsackievirus B infection. Also, infection induces mitochondrial dysfunction and increases the generation of reactive oxygen species. And then finally, that miR7-3HG reduces the viral protein gene expression and also apoptosis and cell self-derived islets. And with that, I just want to thank everybody in my lab for the guidance, support, and mentorship, especially from Jeff Millman. I also want to thank my thesis committee for all the support and ideas throughout my journey. And finally, my funding sources for supporting this work. And then, yeah, now I can take any questions.
Great. Thank you, Daniel. This is fascinating work and really interesting coming on the heels of the PRV 101 from Provention. That's the vaccine for coxsackie-VB, and it's it went through the Provent trial just for safety. So this is really interesting. It adds a whole level of mechanistic understanding of what actually goes on with CVB. And I know we talked a little bit before, but I was thinking, I know Jing Hughes is right down the hall from you guys, and I'm a huge fan of her work. It's really interesting to me that coxsackie-VB does impact primary cilia in the body, and particularly in the airway epithelium. And it causes basically a loss of cilia in the airway epithelial cells. So, and I guess it was also kind of like it disrupts the polarity of those cells and has a tight junction barrier disruption and loss of the cilia. So
Mhm.
just kind of thinking about that and this and what you're looking at here with the CVB infection and resulting changes in genetic changes, basically up and down regulation. What can you say about sort of that information and those facts as it might pertain to beta cell primary cilia?
No, yeah, thanks a lot for that question, Monica, and for the observation as well. So, you brought up Dr. Hughes's work, and they've showed that when you knock out primary cilia and beta cells specifically, you actually see a reduction in insulin secretion. Now, our data set here we didn't have necessarily a ding for cilia gene ontology terms or anything like that, but we did observe a lot of changes of genes associated with a cytoskeleton. And a lot of these are tight junctions, like the one that you mentioned, and other components of it. And as we also know, cilia highly relies on the cytoskeleton. So it's definitely possible that coxsackieviruses can negatively impact the cilia and the beta cells, and that may lead to beta cell stress and dysfunction. But that's definitely something that we haven't looked at yet, but something interesting to look into.
Yeah, I mean, you've got a great setup there to kind of continue the work and look more carefully in that zone. You've got some here in the chat. Fantastic talk. Does mitochondrial transfer between cell types occur in the pancreas as shown between adipocytes and macrophages? From Larry Spear.
Yeah, no, thanks, Larry, for that question. So we don't have any evidence from our data set showing that there's mitochondrial transfer, but I mean, it's a possibility for sure. I think especially when we see the changes in the vesicle-associated genes and other genes associated with protein transport and trafficking. And but I do think one of the things that may be happening within the pancreas itself is the cells are trying to survive. They're trying to meet their metabolic demand, and they can do this in different ways. And my hypotheses, for example, for the data that I showed from electron microscopy, showing that alpha cells increase the mitochondria area, and then beta cells, sorry, the other way around. Alpha cells decrease the mitochondrial area, and beta cells increase it. I actually think that's a mechanism to try to keep up with this metabolic demand. And there's been work shown in other viruses that proves that to survive and decrease, for example, reactive oxygen species within the cell, the cells will induce mitochondrial fission to shrink the mitochondria, or induce mitochondrial fusion to enlarge it. And so I definitely think this is something that's happening within our cells. And we just need some follow-up studies to show that.
That's great. Good answer. Last call for questions. We got a little bit over, but it's really such a fascinating piece of work. Last call for questions. Anyone? All right, then. Daniel, it's really a lovely talk, and congratulations on this work, and best of luck and next steps. We'll keep an eye on you, because I think you've done some really beautiful work here, very detailed and innovative, I'd say. So thanks again for joining us.
Yeah.
All
And
right.
thank you, and to the Sugar Science for the opportunity to talk today. I appreciate it.
Absolutely. Have a great rest of your day.
Yeah.
Bye-bye now.
You too. Take care.